So my name is Shreyas and I'll be explaining how muscles contract and relax. So so you might have wondered uh, how do muscles even move? Like there must be some biological mechanism that is responsible for this, and how are we able to control it on nerve response? Isn't that quite amazing? So yeah, we'll explore that. So let's first have a look at the structure of these muscles. So our muscles are made up of muscle fibers, which can be seen here, and these muscle fibers are in turn made up of myofibrils. So these myofibrils are composed of repeating units called sarcomeres. Now these sarcomeres, let me zoom in. Okay, so these sarcomeres are made up of, so this unit from here to here is like one sarcomere, and these units are made up of I bands and A bands. Now I bands are composed of the thin filament of the muscle, and A bands are composed of the thick filament. So the line bisecting the thick filament is called the M line, and this will be called as the Z disc, the line that bisects the thin filament. So what is the role of the thin and thick filament? So this is a more detailed view of the sarcomere. As I said earlier, this is the Z disc and this is the M line. So how muscles contract is on nerve response by some mechanism which I'll explain later. These thick filaments somehow pull the thin filament inside. So the Z disc now comes closer to the M line, and basically this entire muscle contracts. On the relaxation, the opposite happens. This Z disc is pushed away from the M line, and hence this muscle relaxes. So when the muscle contracts, basically what is happening is the overlap between the thin filament and thick filament is decreasing with the because these are sliding over one another. So let's have a look at the structure of the thick filament. So the thick filament is composed of a protein called myosin. Now these myosin units repeat, and this in turn forms the thick filament. So each myosin unit is composed of a tail, which is the carboxyl terminus, and two heads, which are the amino terminuses, or rather this part is called the amino terminus. So these heads actually have a very crucial role later on. Now let's see how. Let's have a look at the structure of the thin filament. So the thin filament, I'll just erase this and explain once again. The thin filament is made up of units of proteins called actin. So these actin units form one part of the thin filament. So each actin unit has an attachment site on which the heads of the of myosin can attach, like this. So yeah, these can attach here. But along with actin, the thin filament also has a protein called troponin. So troponin, and also something else, another thing called tropomyosin. It is tropomyosin. So these tropomyosin proteins are actually these actually block these binding sites on which the myosin heads were supposed to bind. So just one second, these are tropomyosin, yeah. So these block the binding sites on which the myosin heads are supposed to get bounded basically. So initially the configuration of the thin filament and thick filament is something like this. A bunch of actin molecules with their binding sites covered by tropomyosin and a bunch of troponin molecules here and there. So now, and this here is the thick filament. Yeah, this is myosin and this entire thing is myosin and this is actin. So let's come to the part of nerve signals. We know that muscles only move when we want them to, when some nerve impulse triggers it. So what a nerve impulse does is it releases calcium ions. So these calcium ions come to the thin filament, and this troponin protein is actually calcium attracting, or rather calcium binding. So when troponin binds to calcium ions, a change of conformation happens, which in turn leads to the tropomyosin moving away, and this now exposes the binding sites that were supposed to attract the myosin heads. So this will transform to something like this now, with the binding sites exposed. So now naturally what will happen is the myosin heads will get attracted to these binding sites. At this point, just take my word for it right now, I'll explain later how this happens. ADP and phosphate is present on the myosin heads. So when this myosin head binds to these binding sites, the phosphate leaves. So basically now the situation is something like this. Alright, and now the phosphate has left and we're left with ADP here. This phosphate leaving actually triggers a conformation change, and that causes this myosin head to change angle, and in this process the ADP also leaves. Now understand the significance of this conformation change. This kind of triggers a power move which pushes the actin to one side. This will, okay, just to give some clarity. This will push it to the left side, and hence the actin will move here to the left, or whatever side it moves to. Basically what is happening is the actin is sliding over the myosin, and this is responsible for muscle contraction. So once this moves, yeah, 1 second, so I'll just draw it. It will be something like, sorry, I'll just make the angles a little better. Yeah, so it will be bent and it will still be bonded to these actin molecules, and now the actin has shifted a little to the left. So you might wonder that this happened only once, right? There will be like a very minimal movement due to just one conformation change. But there's a twist. This actually is a cycle. This happens again and again. Now, how? So, you remember that we had ADP and phosphate here earlier. Now, ADP and phosphate are, if you think about, if you like look carefully, they are products of hydrolysis of ATP. So somehow there must be an ATP involved here, and that's accurate. And ATP is involved here. So after this ADP leaves and this conformation change happens, after like the actin is moved, and an ATP molecule now comes. This ATP molecule binds to the myosin head, and that somehow that breaks the bond of the actin and myosin head. So this now separates. After separation, it will look something like this. This is still shifted one unit to the right, left sort of, and now this is separated with the ATP here. Now this ATP, once the myosin head has separated, is hydrolyzed. Now on hydrolysis, this forms ADP and phosphate. Here also ADP and phosphate. And now once again this so now once this hydrolyzes, this again triggers a conformation change in which this angle goes back to normal again. So basically from a bent thing, it will become sort of straight again, like it was earlier. So I'll just label this. These two were the actin molecules that had shifted earlier. Now it is something like this. With ADP and phosphate here, the myosin heads can now bind to a different set of actin molecules, and this basically is the initial step of the cycle. So as long as the nerve signals are coming and the tropomyosin have exposed the binding sites, these cycles will keep on repeating, and hence the muscles will keep on contracting. And so this basically translates to, as long as we're giving nerve signals for muscles to contract, they'll keep contracting. And this is an amazing phenomena, because something as like complex as muscle contraction and movement is understood by such biological processes. So yeah, that was basically how actin and myosin are responsible for muscle movement. So yeah, keep enjoying science. Goodbye for now.